Hi everybody, my name's James Stovold. I'm going to be talking today about my paper Identity Increases Stability in Neural Cellular Automata. So I'm going to assume that everybody knows what a cellular automata is at this point, and for the interest of time, I've not really got time to go into the detail of it. However, some of you may not be aware of what a neural cellular automata is. So whereas in the classical cellular automata, every cell is basically either alive or dead or some discrete number of states, in the neural cellular automata, every cell has a vector of floating point values. So you can see down the left here, we have in this scenario 16 floating point values for every cell. These are interpreted in the following way. So we would interpret the top three as our red, green, and blue channels so that we can produce colored shapes, and then an alpha channel which affects the transparency of how we interpret of what we actually grow. It's also used then to decide whether a cell is mature enough to grow a new cell, so it's used to control some of the growth. Then there are 12 hidden channels underneath. These are for that cell-to-cell communication, so they can store what they want in there and it will be passed to their neighbors at each time step. Because this is now, a floating point vector, we can apply a neural network. So instead of the standard kind of neighborhood rules, which we get in the hard-coded kind of elementary cellular automata or game of life, we use convolutional neural networks. So we can apply a convolution to get our neighborhood, which gives us what we call the perception vector, which is just three of these which represent everything going on around the particular cell, and we do that for every cell in the environment at the same time. We can then pass that through a fairly simple small neural network to get an update for the particular cell we're interested in. And then we apply some stochasticity there so that it doesn't rely heavily on everything updating at the same time, so some cells will change at a certain point. And then we do some masking to make sure we don't get spontaneous growth in random places that has to split out from an existing cell. One of the reasons why we're interested in these, however, and the original idea of this was that it's a model of artificial morphogenesis. So while these have been applied in a bunch of different interesting areas, like creating interesting dynamic textures, what I'm interested in is actually growing organisms and seeing whether or not we can study those organisms. A couple of years ago in Japan, I presented some work which looked at how we could adapt NCAs so that the organisms we grow can actually respond to different types of signals. So I identify kind of three groups of signaling, and we focused in that work on the internal and the external signaling. So that's kind of internal to the organism, so that's something like a hormone or a genomic signal, or external, i.e. something coming from the environment. And I kind of waved my hands a little bit and skimmed over societal signaling because, well, it's really difficult. And so societal signaling was when you've got multiple organisms which then need to interact in some way. But that's what I've now been focused on for the last 2 years, trying to get that working, and turned out it's a lot more difficult than even I originally thought. But we're going to give it a go. Okay, so social signaling is problematic. It poses a real issue for NCA organisms because, as you can see in the animation on the right there, when we grow two organisms very close to each other, they don't realize that the cells that are next to them are part of a different organism. They just see a cell. And so you get these really interesting but irritating effects, like kind of extra heads growing out of them or extra legs, because they don't understand that that is not them. They're just applying the update rule. And so where we want to have multiple organisms near each other and communicating in some way or producing a signal, sending it to another one, we need to fix this problem. And that's the focus of what I'm going to present to you today. So I had this idea of maybe I could apply some work which I had done previously. So back in 2014, I presented some work on emergent identity formation in swarms. I was looking at Boids at that time. And what I did was apply the Firefly synchronization algorithms, just coupled oscillator, basically. And in that algorithm, effectively, as you're counting up to some threshold, you spike at that threshold or you flash at that threshold, and you just adjust according to what you see around you. The idea was, what if we could put this Firefly algorithm into the NCA as it's training? Can it train to work with this algorithm as well? Turns out, no, doesn't work. One of the main issues here is that the identity which it's learning or creating is changing all the time because it's emergent from all of the different cells which are in the organism. And the issue with something which changes all the time is the neural network tends to learn to forget about it and ignore it because it's not giving it anything useful. But I had an idea off the back of this, which did work. And so the idea of that was, instead of using the Firefly algorithm in training, what if we just trained it with what we expect the Firefly algorithm to produce, and then apply the Firefly algorithm or the emergent identity algorithm in testing? So round two, what I did this time was, instead of just actually running this algorithm, the identity algorithm, during training, we just said, okay, just reproduce this value. I will come up with the identity for you. For now, remember this number effectively. And this extends this signaling work because the internal signaling is basically doing exactly this. Except this time what we're going to be doing is asking it to remember that number rather than just using it to guide it to a particular attractor. So how do we go about doing this? Well, on the left here, we have the diagram you saw earlier. I've extended this to an extra channel. So I've just put now what I call an identity channel at the bottom here, which is just like any other channel, except I'm going to include that in my loss function. Okay, so I'm going to guide the training with that in there. So it basically just gets pushed onto the bottom of the RGB values, and then I can use that as a way of providing a loss signal. It's worth pointing out, because this is something which makes what I'm about to show you even more interesting. During training, the organisms are completely isolated. So just like in the original model, they don't see any other organisms other than themselves until we get to the testing phase. So they have no concept of other organisms. That all the cells they've ever seen have been part of themselves. This is important, and you'll see why when we get to some of the results. Bear that in mind. So we trained 3 different models. Model A is just a control case, the same as the original NCA, except we've got the extra channel there. We basically just don't put any restrictions on that channel. We train it just like the original NCA, but we didn't want some confound of there being an extra channel, and that's why the other models were there. So we used 17 channels here. We trained model B. So this one, we trained it to produce the value 1 on the ID channel of every living cell. Simple as that. Just when we do the loss function, we include the ID function and we make that entire layer of 1s for every living cell. If it's a dead cell, it doesn't pay attention to it anyway. And then model C is effectively the same as model B, except instead of just remembering the value 1, we give it a value on the seed cell, and it has to remember that value. So it has to propagate that as it's growing. And that can be either 0, 0.5, or 1 in the training which we did. This is very close to what we were doing with the internal signaling before, except now it's actually having to reproduce that value, whereas before it wasn't. So how do we test what we're doing? So we're not even at the stage now where we can use the Firefly algorithm. That's still going to be put into future work. For this, what I wanted to do was to test how does this affect the behavior of the organisms. So for each of these 3 models, A, B, and C, we grew 2 organisms in close proximity, and we kind of varied different parameters so that they interact in different ways. So you can see on the right here what I mean. So the gecko emoji can be put in a variety of different things because it's non-symmetrical. So we've had to move the seed cells around a little bit in order to get different interactions and different amounts of interactions in different places. We let all of these organisms grow for 1,000 time steps. Then we took what they had grown into and compared it to some idealized images. The ones on the right here are idealized images. And when I say idealized, it basically means I've taken the target image and just put 2 of them on top of each other. And you can see, especially over here, some bright green patches. This is where they overlap, because it's additive. You've got this very bright chunk where they overlap. So we're always going to get some deviation from ideal, but it doesn't really matter for what we care about. And the 3 parameters we looked at were the lateral distance between seeds, so that kind of horizontal distance, the vertical offset in both directions, and then the difference in seed time, so which one grew first and for how much of a head start it got. So is it a fully formed organism before the second one grows, or are they growing at the same time? So into the results. So it turns out, and you can see this here, and I'll talk you through this graph in a second, introducing this identity value really increased the consistency and the stability of these organisms. So we can see on the graph here, we've got three panels. Model A is on the left, Model B in the middle, Model C on the right. So Model A is our control case, which has none of this identity. The other two have different types of identity in them. Each of these values across the bottom here are the lateral distance. So they're getting closer and closer as we go towards to the right. Okay, so that's why we're seeing an increase in error. And you can see up the vertical axis here, this is our error. Error is not really the best term to use here. It's basically difference from that idealized image. We're just using root mean squared error because it's convenient. And we can see just by taking sort of a wider view of it, that there's a lot more consistency in B and C than there are in Model A. Model A, very much straight away, we start to get things breaking down. We start to get this tumor-like growth, which we all know and love from the NCAs. Much less consistent in its behavior. You know, the range of values we're seeing here is much larger than we see, for example, in Model B or Model C. Now, the paper goes into details around how much difference there is and everything like that. I'm going to skip over that for the sake of time. If you're interested, feel free to have a look. What is interesting is that Model B and Model C behave slightly differently. And this was a bit weird, and I was trying to understand why this happened, and that's the focus of basically the rest of what I'm going to talk about today, assuming I have time. So if you look at, for example, 9 and 6 here, you can see that the behavior between Model B and Model C is actually quite different. It's not as different as Model A, don't get me wrong, but there's something different happening here, and I wanted to understand what that was. So this graph on the left here is the same data as before. I've just reorganized it, so I've got rid of the larger distances because they don't interact that much, so we ignore them. And I've just got the panels are now distances so that we can directly compare the behavior of the different models. And you can now see much more clearly that B and C behave differently, especially at distance 9. Distance 12, this is more what I would expect. Some error still there, some variation, and mostly just that's the difference between the idealized image not being ideal. But if it was just the idealized image, then B and C should be the same as each other. It would be different to A, but not to each other. So there was something very strange going on here. So what I did was basically write a little bit of code which found me the ones which had the biggest discrepancy. You know, something where Model C was doing something very weird and Model B was behaving itself, which is what we've got here. And so what we've got on the graph on the left here is the error value over the growth of the organism. So you can see there's a massive drop at the beginning as it goes from basically nothing to growing into this shape. Model B then stays very low and very consistent, and Model C starts to drift. And what we're going to see on the right is this animation of the growth of Model C in this case. So we see the two organisms grow. And what you can see is the organism on the right, or on the top, starts to shuffle away from the one underneath. And that's this kind of uptick in error which we're seeing. What's going on here is that the organism is growing fine, and it's able to keep itself consistent and stable to the point that it's able to move away as an organism from another organism. And this happens in a bunch of different directions, and is completely unexpected from the perspective of what we've trained it to do. You know, these organisms have never seen another organism before now, and yet they're now able to shuffle away and become more consistent and stable as a result. A reasonable question would be, is this just Model C? Well, Model C tends to move more than Model B. What we've got here is actually just a bounding box change, so the position of a bounding box around all the living cells. Again, I'll let you look at the paper if you're interested in this because I'm running out of time. But Model C does seem to move more than Model B, is the bottom line of this. So to conclude, we can stabilize NCA organisms by just asking them to remember a number. When we do that, we can then see emergent behavior when we put them close to each other and we train them, we grow them close to each other. And this seems to be more pronounced when more identity values are there, although we're not entirely sure why. future work, still trying to get social signaling to work, organism to organism interaction, and possibly some look at the links between identity and individuality from an information theoretic perspective. So thanks for listening, and yeah, that's my work.
My name is Akarsh, and I'll be presenting our work on automating the search for artificial life with foundation models. So as you guys know, artificial life has many simulations ranging from like evolving 3D block creatures to simulating low-level cell-like dynamics to even simulating ecosystems, or the most exotic one I like is the self-replicating programs, right? And these are all super cool and great, but they have a downside in that most of these simulations were hand designed by humans in order to get interesting behaviors to come out of them. So this begs the question is like, as A-Life simulations get more and more complex, do we really want to keep tediously hand designing this simulation rules and parameters in order to get interesting behaviors? I have a thought experiment. Imagine trying to hand design the periodic table of elements and the pairwise interaction between all of the elements in order to get life to emerge. I personally wouldn't want to be the one doing that. And if you think about it, if the simulations we're creating are truly going to be emergent and open-ended and surprising, then by definition, we can't really rely on intuition to design the rules of the simulations, right? So we clearly need some way to automate the simulation design and search. Basically, what we need to do is a search for simulations which have good properties or are interesting to a human. So in order to do this, we really need an objective. And one option that we have is just to try and mathematize interestingness or open-endedness or emergence. And certainly people have tried doing this, but the downside to this is that they almost always have a misalignment between what the equation says and what a human really means by interestingness. And this is a big deal when you're optimizing for something because you get something that you asked for, but not something that you wanted, right? So another option we have is just to ask a foundation model. And this may seem naive at first, but if you, the more and more people are realizing that as you make these models bigger and better, the way that they see the world more and more aligns with how a human sees the world. And this gives us a good grounding in which the foundation model should be a good proxy for a human in this situation. When that brings us to our work, ASIL, Automated Search for Artificial Life. So at a high level, what we're going to do is basically take simulation configurations, run those simulations, pass the results into a vision language foundation model, and somehow extract a score from that, and then use that score to find better simulation configurations and thus find better simulations. And we propose three ways of using ASIL, basically made from using supervised target, using a natural language description of what you want, the search for open-endedness, as in you're finding temporally novel simulations, and illumination, basically finding a big set of diverse simulations in this space of simulations. So I want to start by reformulating the problem. So instead of designing a single simulation, what we're going to do is allow the researcher to define a more generic set of simulations. And this is often much, much easier than designing a single specific simulation. And basically, you can imagine this is the difference between designing that periodic table that I talked about versus just specifying that there's going to be 100 elements and there needs to be a 100 by 100 interaction matrix and letting the algorithm find the specific interactions. That's a much, much easier problem, right? And what we're going to do is we're going to parameterize this entire space of simulations using theta. And theta is basically going to describe the initial state distribution of that simulation, the dynamics rule for that simulation, and the rendering rule for that simulation, which takes the state and renders it as an image or anything that can be inputted into the vision language model. And then all we have to do is just search for what we want in this space. So let's talk about how we're going to do this for the supervised target via natural language. What we're going to do is we're going to take our theta and we're going to sample an S naught, like the initial state, and we're going to step this initial state t times, like we're simulating the world for t time steps, and then take this final state and render it using the render function. And together, this is going to be our RS function. And what we can do then is search for the theta such that when you run the simulation using the RS function, then you're going to end up with a sequence of images. And from the sequence of images, you can take each image and embed it using the vision language model's image embedding function into a latent representation space, what we call the foundation model representation space. And basically now you have a trajectory of latent points in this space. Then what you can do is you can basically measure how close this trajectory lines up with what the thing that you want, which is the trajectory of prompts that you can describe. So in this case, we're basically measuring how close a simulation is to going through the points a cell, two cells, and then an ecosystem. So in this way, we're basically quantifying how much does this simulation entail like self-replication, right? So let's get into some more details in this then. So the actual image embedding and the text embedding, what we're going to do is use CLIP. And CLIP is just basically a giant AI foundation model trained by OpenAI that jointly trains the image encoder and the text encoder. It's perfect for what we exactly need. And then for the actual argmax, the optimization, we're going to use CMES, which is just a black box optimization algorithm, which is going to try to find simulations which maximize our score of what we want. And then if you run this for 3 classes of problems like linear, Boids, and particle life, you end up with some really intriguing results, which I'm going to get into now. So for Linear, as you know, it was made by BERT. It generalizes Conway's game of life to our continuous RGB values, and each of the cells in this grid use the same local differential equation update rule. And what we're going to be doing is just searching over the initial state dynamic and the initial state and the dynamics parameters of the simulation. And if you do this, you can see that we can find really aesthetically pleasing results. You can search for a simulation which looks like a multi-celled organism or a caterpillar or even a pepperoni pizza. My favorite is the self-replicating pattern because when it runs again, you'll see that it's literally starting off from a small patch and it's outwardly growing a self-replicating pattern. Next up, we can do this for Boids, where we simulate 128 bird-like objects which move through 2D space, and each Boid basically only sees its local neighbors and decides whether to steer left or right based on what their local neighbors are doing. And all of these Boids in the simulation share the exact same neural network, and what we're going to be doing is searching over the neural network parameters. So this is basically like an ant colony where you're optimizing the behavior of one ant such that the global behavior of those ants when you run the simulation is interesting to that foundation model. And you can see we can find simulations which look like a snake or a Fibonacci spiral in nature or even collective intelligence. And it kind of just works, even in this very non-differentiable, very weird domain. For the mapping from parameters to the actual final image is very nonlinear and chaotic. We can even do this in particle life where we're simulating 1,000 particles in 2D space and there's 6 types of colors or 6 types of particles, each designated by color. And the color basically determines how the particle interacts with other types of particles, right? And what we're going to be doing is just searching over the 6x6 interaction matrix, which dictates the physics of this world. We're going to search for physics, which self-organizes into certain behaviors that we want. So in this case, we can find concentric circles or a caterpillar or even like a red organism, and it kind of just works. Another thing we can do is try to search for temporal targets. So in this case, we took a neural cellular automata and optimized it with SGD and backprop through time. And what we did was we basically just optimized the first half of the simulation to look like one biological cell and the second half to look like 2 biological cells. And you can see that it kind of nails it. It takes that cell and it has it split into two in order to create a self-replicating pattern, sort of. And the final way we can, I guess the second way we can use our ASIL is for illumination, finding a diverse set of simulations. And what we're going to do is basically instead of finding a single simulation, we're going to look and try to find a set of simulations such that when you run each simulation independently using the RS function, they produce images which map to a set of trajectories in the latent space. And what we're going to do is try to take this set of trajectories and push them apart. And we're going to try to find basically a set of simulations which are diverse in this foundation model representation space. And if you do this for Linea, you end up with a lot of very, very cool looking diversity, and it seems to be working really well. And we actually ran this with 4,000 simulations. So we found 4,000 diverse simulations. And what we did was we asked Clip to basically take their Clip vectors and project them into a 2D UMAP and then plot the simulations in this thing, what we call a simulation atlas. And you can see Clip does a good job of organizing the simulations based on shape and color into really the things that matter the most. So you can see on the right, we have the red ones, and on the top, we have the blue ones, and the shapes are also organized in such a manner. We can do, and here's a larger simulation atlas. We can also do this for Boids, and what you can see is that here's a larger set of Boids. We can make a simulation atlas of Boids, and you can see that a clip organizes them in a way where on the left it's all of the dotted Boids. On the top, it's the flocking Boids. On the right, it's the sparsely flocking Boids. On the bottom and bottom left, it's the circling or the snaking Boids patterns. And these are basically all the different behaviors you'd like to see from the Boyd's simulation, and our method is able to discover all of them automatically. And here's a larger simulation atlas of Boyd's that you can find in our paper. And one thing I want to point out is if you do this in pixel space, it doesn't work that well. It basically just creates simulations that look very aligned to a human. But if you do this in Dino space, it works just as well as Clip. So the main thing is just that's doing this heavy lifting is this the foundation model. So the third way you can use our method, ASO, is to search for simulations which are open-ended. And by open-ended, what I mean is that if you take the simulation, you run it using the RS function, you basically want to end up with trajectories in this latent space that are diverging and outwardly diverging. And the way we can quantify that is basically measure for each point in the trajectory, how close is it to the history of points that came before it in that trajectory? And this gives us a way to say how divergent is this in this latent space, right? And how much novelty is it creating? So here's Conway's game of life. If you do our metric in pixel space, it produces just static noise, which is not interesting to a human. But if you do this with clip space, it ends up finding these are the top 4 most open-ended simulations. It finds simulations with a lot of human structure because this foundation model space is so aligned with the human space. And so these are just cellular automata rules which produce interesting behavior, just like Conway's game of life. And here's a visualization of the actual trajectories in the clip space over the simulation time step. And you can see it truly is really much more open-ended than Conway's game of life or the pixel space optimized simulation in clip space. And one thing you can do is just take the entire space of simulations. In this case, there's 260,000 simulations, and just render them as images and pass them into Clip, and then just organize just cluster this entire thing based on how Clip sees this space of simulations. And you can see it sees a bunch, basically 4 different islands of types of simulations. And this is a good way to basically get a grasp on what the entire space of simulation looks like. Another thing you can use these foundation models for is to quantify these previously qualitative phenomena. So in this case, we took one simulation found by ASIL and another one on the right found by ASIL and linearly interpolated the parameters between them. And you can see in between, it doesn't appear to look like the left or the right. And you can use CLIP to quantify how dissimilar they are. So you can turn a qualitative result into a quantitative result using CLIP. You can also measure, you can add more particles to your simulation and see when does a caterpillar begin to start emerging. And in this way, you can use CLIP to basically quantify the Moore's different insight from the seminal physics paper. You can also use it to basically just sweep the parameters and see which parameter is the most important to the simulation and what it's doing. So we identified one parameters in this caterpillar simulation, which basically, if you sweep it, it completely destroys the caterpillar, and CLIP is able to identify that. And this coincidentally turns out to be the parameter that controls the interaction between the green and the yellow, which makes sense. You can also use the clip change in the latent space over time to quantify when a simulation has plateaued, as we did here. So looking forward, I think that you can use ASIL, which is this very general algorithm, to search for your own substrate. So here, what we did was I made my own substrate, which I call particle life plus plus, where basically I allow the colors of particle life to change along with the position and velocity. So a yellow particle can hit a red particle and become orange. And this basically unlocks like this chemistry in this thing. And you can use ASIL to kind of search for open-ended ones here. And I encourage you guys to try ASIL out of the box on your own substrates. Over here, we did something cool where we basically took 16 of the Boyd simulations we discovered, and then after some time, we let these Boyds enter each other's worlds. So you can see that these Boyds are actually going off and like kind of like intrusive species going into other people's worlds and starting to mess with them. We also did the same thing with the game of life, and we found very dominant game of life rules and some submissive game of life rules. And what now? So I think looking forward, I want to answer like what's stopping us from achieving this ultimate dream of ALIFE, which is to recreate the diversity of life on Earth, right? And I tried to do this here where I tried to take an NCA and try to have it go through self-replicating molecules, self-pulling primordial soup, finally getting alien animals roaming around. I think the key things that are stopping us from achieving this is the expressivity of our substrates, the search algorithms, and finally the scale at which we're operating at. I think just like AI, we need to pour lots of resources into our simulations and just increase the scale at which we're operating. And finally, I want to thank my collaborators. As you can see here, this work would not be possible without them. And with that, thank you.
Hey everyone, we are recent master's graduates from University College London, and we're excited to present our work on guiding the evolution of artificial life using vision language models. The project actually grew out of a module on open-endedness and general intelligence we took at UCL, taught by Tim Rocktaschel. What you're seeing here is a quick showcase of our system ASAL++. We started with the simple text prompt and egg hatching, and our method autonomously evolved the simulation by generating its own series of increasingly complex prompts, like symbiotic fungal networks, and eventually mineralized crystalline spore dispersal. This dynamic process leads to some really interesting and unexpected emergent behaviors, which is what we're here to talk about today. The idea of artificial life, or ALIFE, has been around for a while. Many of you have probably heard of Conway's Game of Life from the 1970s. It's a classic example of cellular automaton, which uses a discrete grid, simple rules, and binary states to produce incredibly complex, unpredictable patterns. Our work uses a more modern substrate called linear. You can think of linear as a continuous version of the Game of Life. It uses continuous states and values, which allows for smoother, more fluid dynamics and far more intricate, quote-unquote, lifelike patterns. We choose linear as our primary environment for these experiments. Our work, ASAL++, builds on the foundations laid by Kumar et al. In their algorithm called ASAL, which stands for Automating the Search for Artificial Life. The core idea of ASAL is to use a vision language model, or VLM, to steer an ALIFE simulation toward a user-defined text prompt. The prompt is called a supervised target. The engine for this is CLIP, a model that understands both images and text in a shared embedding space. This allows us to directly measure how semantically close a frame from our simulation is to a text description, simply by calculating the cosine similarity of their embedding vectors. Then, ASAL optimizes the simulation's patterns to maximize the similarity score, effectively finding a simulation that looks like the prompt you gave it. As you can see from these examples, it's a powerful method for discovering specific phenomena, like a caterpillar in linear, or collective intelligence in the void substrate. So while ASAL is a fantastic tool, we felt there was an opportunity to make it more, open-ended. In the original method, all the target prompts are fixed in advance by a human. This predefined objective can limit the potential for true discovery. The theory here, inspired by researchers like Kenneth Stanley, is that fixed goals can cause you to miss unexpected stepping stones. Intermediate discoveries that might unlock completely new and more complex possibilities you never would have planned. So our core idea for ASAL++ was to introduce a second foundation model into the loop. an evolver model. Instead of a human setting the goals, this model dynamically proposes new targets based on the simulation's visual history. This allows the system to lay down its own stepping stones, creating a truly emergent evolutionary trajectory. Let's walk through how that works. Step 1, initialization, where a human gives a single seed prompt, like the Big Bang. Then we enter step 2, an iterative loop. First, we use the original ASAL method to optimize the simulation to match the current prompt. Then, and this is the key part, we feed the video of that simulation to our evolver model. We used Geometry and ask it, quote unquote, what should come next? It generates a new target prompt, which is added to our evolutionary history, and the loop repeats. We explored two main strategies for this. First, EST. Here, the evolver model only sees the most recent prompt, and the simulation is optimized only for the new target. This approach is designed to promote maximal visual novelty and diversity. Second, ETT, Evolved Temporal Targets. In this version, the evolver model considers the entire chain of past prompts, and the simulation is optimized to match the whole sequence. This encourages more coherent and interpretable evolutionary stories, where each stage logically builds on the last. Now, the idea that you can truly quantify open-endedness is still a topic of debate in the research community. For our work, we use a proxy called the open-endedness score, or OE score, which measures novelty within a simulation. The idea is pretty simple. For any given frame in our simulation video, we use clip to compare its similarity to all of the frames that came before it. The OE score is simply 1 minus the maximum similarity found. So a high score means the current frame is visually very new compared to the simulation's history, indicating that novel behaviors are still emerging. We ran experiments giving both EST and ETT the same set of 9 initial prompts and let them evolve. The results confirmed our hypotheses. If you look at the table, EST consistently achieves a higher mean OE score than ETT, suggesting it does in fact generate more novelty within each simulation. More importantly, the OE score difference column shows the changes novelty from the first iteration to the last. We see a statistically significant increase in novelty over the run. For ETT, the score stays relatively flat, indicating it maintains novelty but doesn't necessarily find more of it over time. So there's a clear trade-off. EST gives you greater diversity, while ETT gives you more coherent, story-like evolution. To push the idea of open-ended exploration further, we experimented with generating these trees of life, motivated by phylogenetic trees in biology. To do this at each evolutionary step, we promoted our evolver model to generate multiple alternative next steps instead of just one. We encourage this diversity by setting the model's temperature parameter to 1. This allows us to visualize parallel evolutionary trajectories branching from a single ancestor, like this one starting from a caterpillar. It really highlights how the same stepping stones can lead to completely different and interesting outcomes, strengthening the case for this kind of open-ended search. To wrap up, we've presented ASAL++, a framework that uses foundation models to autonomously generate evolutionary goals for artificial life. Our two trajectories, EST and ETT, offer a trade-off between visual novelty and narrative coherence. The method does have its limitations. We notice a biological bias in the language model, and the linear substrate itself favors circular patterns. But we believe this points towards exciting new directions for foundation model-driven, open-ended discovery in ALIFE. If you'd like to try it out for yourself, please check out our QR codes for our GitHub repository and the Colab demo. We'd like to quickly thank our supervisor Lara Weiss, as well as Akarsh Kumar for his feedback on our original work. Thank you for listening, and now we'd love to show you a live demo. The Colab here in the live demo can be used to simulate EST and ETT given the prompt that you want to set initially and any number of internal evolutionary loops. The temperature, the max number of images from the simulation video you want to give, so on and so forth. And from this, we can start to see some different evolutions of different things. I hope you enjoyed listening, and now I'd like to thank you, and bye.